Everybody's doing wonderful. David Juarez-Flores, who is at Colgate University, a rising senior in the McDonough lab. Don't know.
Morning, everyone. Thank you, Dr. Ellenbogen, for that introduction. And today I humbly am able to present my work that I was able to do here over the summer at UW with the great mentorship of Dr. McDonough. And essentially, it is a pilot study of hippocampal microglia morphology in a piglet model. So for some background, we've seen in observational studies of premature infants that they have higher rates of cognitive, behavioral, and motor deficits. In tandem, they also have high infection susceptibility, underdeveloped intestinal barrier, which could lead to an increased risk of systemic inflammation, which could in turn negatively affect neurodevelopment. The same observational studies have shown that diet affects the neurodevelopment of premature infants. However, the mechanisms of this are unclear. However, this is the limit we can take human models, and the next best model is a pig model due to its high genomic and anatomic homology, as well as similar developmental trajectory to humans. And so the overarching hypothesis of our study is how the food source can affect the development of the intestinal barrier, which that will in turn affect wider systemic and intestinal inflammation. That will in turn play a role in neuroinflammation and early neurodevelopment. And so to begin with the methods, our colleagues at the University of Idaho nurtured these pigs on either human milk or infant formula for 4 weeks. Upon the 4 weeks, pigs were sacrificed and tissues and organs were collected, and we were sent the brains. When we received the brains, we used cryosectioning to set. Okay, sorry. We used cryosectioning to section the samples thin enough to be able to analyze cell morphology later on. And in our case, cell morphology is important because to assess neuroinflammation, we have to talk about microglia. And what microglia are is they are the resident immune cells of the central nervous system. They are also the first responders to any inflammation cues. And the way they respond to inflammation is through their activated state, wherein they have changes in morphology. Typically, they exist in a surveying state where their processes are more ramified and they extend to wider reaches of the brain. And in a more amoeboid state, they are more simple and exist in a more amoeboid state. So assessing these changes in morphology allow us to infer the activation state in the brains. And to be able to see the microglia, we use immunohistochemistry, which is used to label and visualize the cells. It consists of a primary antibody coupled with a secondary antibody paired with a fluorophore. And IBA1 is used to label the microglia, its soma, its processes. Chi67 is used to label proliferating cells, which is another aspect of microglial response to inflammation or damage. But that is not the focus of this study. And DAPI is used to stain the DNA and the nuclei. And so I sampled randomly from the hippocampus of the pigs. I collected random points enabled to be able to assess the cell morphology later on. And I used these random field of views, such as the one on the left, and ran it through a MATLAB script called 3D Morph. And what 3D Morph essentially does is it skeletonizes the microglia and analyzes it on various parameters. And here on the right, you can see how it can skeletonize a microglia, and you can see its branch points and its endpoints and the way it branches out, and it can shoot out data for us, such as territorial volume, cell volume, average branch length, number of branch points and endpoints, and etc. And we could use this data to assess the cell morphology. And I had an N of 1. So I had one brain from a pig that was raised on human milk and another one that was raised on infant formula. And upon analysis of the data collected, it was shown that the microglia of the piglets raised on human milk had a higher territorial volume in comparison to those raised on infant formula. And what territorial volume is, it is the volume of the brain that is reached by the processes of the microglia. So essentially, the processes of the human milk group extended out more. They were more ramified. And they also had longer average branch length, as well as a higher number of branch points. And these work well with the territorial volume, because this can begin to suggest that the human milk microglia are more ramified and complex in comparison to those of the infant formula group. And this suggests that they are more mature and exist in a less activated state and in a more surveying one. And what this can begin to have beginning evidence towards is that diet may affect the activation state of the microglia, which down the line may have an influence on the neurodevelopment of the pigs through the gut-brain axis. And this data set is not complete. I had an N of 1, but the initial pilot data set is an N of 3. So yeah, those are in the checklist for later on. Other metrics also of the microglial phenotype can be analyzed in future studies, such as cytokine expression, as well as transcriptomic state. Different regions of the brain as well, such as the amygdala and the cerebellum, which are connected to the hippocampus, are also on the checklist, as well as white matter development, such as myelination and oligodendrocyte maturation, which are key aspects of postnatal neurodevelopment. But I'd just like to take a moment to appreciate Dr. McDonough for all her help, all her mentorship and patience, as well as Dr. Weinstein for his co-mentorship, Dr. Ellen Bogan, and Dr. McDonald for this wonderful opportunity, and Julie for her wonderful coordination throughout the summer, and the residents, the physicians, the surgeons for the wonderful opportunity and shadowing, and to everyone else involved. But thank you.